The work I will present today is domain promiscuity correlates with rates of domain gain and loss. Here shows the multi-domain protein, the insulin receptor, as an example. The domains, as sequence fragments encoded, are labeled in colored shapes. The corresponding 3D structures are also shown at right-hand side. Protein domains vary greatly in their promiscuity, their propensity to form different combinations with other domains in the same protein. For example, this figure from the literature shows the SH2 domain in gray, with many different domains it combines with. While some domains appear in many combinations, others are observed in only one or very few combinations. What determines domain promiscuity remains an open question. Domains may be promiscuous because they are mobile, or because they facilitate the evolution of novel protein functions. Here, we investigate the potential correlation between domain promiscuity and domain mobility, using estimated domain gain, loss, and duplication rates as a proxy for domain mobility. In this work, we looked into the genomes of 19 metazoan species and 2 unicellular outgroups. The data of 1283 domain families across all genomes are downloaded from the superfamily database. With the domain family dataset available, we can infer the domain family event rates. We can also obtain promiscuity values on those domain families. And then we'll be able to compare between domain event rates and domain promiscuity. Previous works have used parsimony models to link domain events and domain promiscuities, which gives a lower bound on the number of events. Here, we infer the domain family event rates using a probabilistic birthday scheme model implemented in the software count, giving the species tree and domain family abundance in present-day species as the inputs. The rate parameters that best explain the present-day family sizes were inferred using maximum likelihood estimation. In this model, rates were allowed to vary across both lineages and domain families. And here, we will focus on family-specific rates only. Now that we have the event rates available, how do we measure domain promiscuity? Different promiscuity measures have been proposed in previous works. Here, we focused on the number of different domains that co-occur with a domain, the number of bigrams that contains the domain, the number of trigrams that contains the domain, the weighted bigram frequency, a measure of relative entropy. Because promiscuity is limited by the number of times a domain appears, we also considered two measures of domain abundance, the number of times the domain appears and the number of proteins that contains the domain. The size of domains has also been suggested to affect mobility. Therefore, we also considered amino acid sequence length for each domain. To assess the agreement between those various promiscuity measures, we calculated pairwise Pearson correlation coefficients. We observed that promiscuity measures are strongly correlated with each other. Promiscuity measures are strongly correlated with abundance, and domain size is not correlated with either promiscuity or abundance. Now we are ready to ask whether domain promiscuity is associated with mobility. We used a linear regression model to determine whether domain event rates are predictive of domain abundance and domain promiscuity, and the significance of those relationships. Same analyses are repeated for domain length. Coefficient values from regression results show that abundance and promiscuity are positively correlated with gain and duplication rates, negatively correlated with loss rate. Meanwhile, contrary to suggestions in the literature, our regression results suggest that there is not enough supporting evidence for the proposed hypothesis that smaller domains are more mobile. Because domain promiscuity is bounded by domain abundance, we want to know whether the significant correlations observed were byproducts of dependence of abundance on event rates. For that matter, we included domain abundance in the model as an independent variable. When abundance is included in the regression, coefficient values show promiscuity is strongly dependent on abundance. New results also show a weaker, less significant correlation with gain and duplication rates. Moreover, promiscuity is not correlated with loss rate anymore. We found that mobility is a strong contributor to domain promiscuity because mobility increases domain abundance. And controlling for abundance eliminates most but not all of those contributions. We also observed no relationship between domain length and domain mobility or domain promiscuity. Thank you for your time and interest.